Namaskar children, in the previous class we had started with the chapter Heredity and Evolution. Then we discussed about the definition of heredity, and then we discussed about the accumulation of variation during reproduction. Now what will happen, as we know that variations are useful for the species, it is not necessarily useful for the individual organism. Then we discussed about the inherited traits. In that we discussed about Gregor John Mendel, the father of genetics. Okay, he took 7 characteristics of the pea plant, and why did he chose pea plant, that also we had discussed. Now today we will be talking about Mendel's law of inheritance. Okay. Now let's talk about the first law, that is law of segregation. Mendel's first law of inheritance, or the law of segregation. The characteristic or trait of an organism are determined by internal factors which occur in pairs. Only one such factor can be present in a single gamete. Now if I talk about the trait, it means each trait can be influenced by both paternal and maternal DNA. Thus for each trait there will be 2 versions in each child, as we have discussed, that if I talk about height of the child, it is being influenced by 2 traits, and the dominant trait will only be expressed. For the hair texture, there are 2 traits. One is coming from the maternal side, other is coming from the paternal side. It is being influenced by 2 genes, but at the end it will be expressed. Either it is, codominance, either only a dominant gene will be expressing itself. The trait seen in the child will be what? Depending on what? Depending on the dominant gene or the codominance. So here if we see, they have taken one example. Here we are talking about only one characteristic, and that we will consider as monohybrid cross. But now we are not talking about the monohybrid cross. We are trying to discuss, what is exactly happening here. If I talk about the tall plant, you can see in this picture, it is being represented by capital T, it's pure tall plant. Now if I talk about the short plant, it is small t, pure dwarf plant. When there is a cross pollination between them, the resultant plant will be tall. You can see here this plant is also tall, but here the situation has changed now. Here the genotype will be capital T, small t. No doubt, small t, the dwarfness is not being expressed, because for dwarfness to be expressed, or for recessive gene to be expressed, it needs 2 copies of it. It needs small t, right? But it is being carried now, and you never know in which generation it will be expressed. So when you crossbreed these 2 plants which are having genotype capital T, small t, you can see this is the cross, capital T, small t, the resultant plant will be capital T, right? One plant will be capital T, then capital T, small t, that is 1:2:1, right? So here if you take the proportion, you can calculate what will be the ratio, and one plant will be small t. That means this one plant is pure tall plant, this one plant is pure dwarf plant, and that is why dwarfness is able to express. Whereas in these 3 plants, they all are tall, but if you check the genotype, it is different. Okay, I hope you have understood. Okay children, now let's start with the monohybrid cross. When plants having one pair of character that we have discussed, we are going to take tall and dwarf. For example, tall and short plants was crossed, that's monohybrid cross. Let's see what will be the situation. Mendel selected pea plants having one pair of character, a tall pea plant and a short pea plant. He selected pure tall, that is capital T, and pure short, that is small t, pea plants, and cross pollinated them. As I told you in the previous slide, pure tall plant, pure dwarf plant. He obtained all tall plants. Here the phenotype is what? It's all tall plants in F1 generation. But what is the genotype? Has it changed or has it remained the same? Yes, it has changed. Now instead of capital T, we have capital T, small t, right? In the first generation. When the first generation plants were self-pollinated, that's very important point. First of all, you are supposed to cross-pollinate them. In F2, to get F2 generation, you need to self-pollinate them. He obtained tall and dwarf plants in the ratio 3:1. So, if I talk about 100 plants, right, 75 plants will be tall and 25 plants will be dwarf. Okay, 3:1 in the second generation, that's F2. The ratio of pure tall, that is capital T, hybrid tall, capital T, small t, and pure dwarf, small t, was in the ratio 1:2:1. Are you getting my point? Now 1:2, that means they all are tall, but here if we talk about the genotype, it is different. Okay, the trait that is expressed in the F1 generation is called the dominant trait, and the trait that is suppressed in the F1 is called the recessive trait. You can see here also the diagram is given are round seeds, wrinkled seeds. Can you see this, children? Yes, this is the second characteristic. You know, in the examination it can be this also, it can be tall also. It out of 7 characteristics, any characteristic can be taken, but the cross remains the same. Remember, here you have to write in steps. I will tell you how to write. I will dictate at the end how to write in the steps. Then here you can see this capital R for the round seeds, small r for the wrinkled seeds, because this is the recessive trait. Then in F1 generation we will get seeds which will be round in shape, but now the genotype has changed. It is now capital R, small r. And then in F2 generation it will be what? 3/4 of the seeds will be round, whereas 1/4 only will be wrinkled. So, we will get 3:1 ratio. So, first we have to do cross fertilization, and later on we have to do self pollination or self fertilization. Okay. Now, you can see on the same slide this first diagram, short pea plant and tall pea plant. Cross pollination is done here, and we will get all tall pea plants. Then when you allow them to self-pollinate, out of 4 plants, 3 plants will be tall and 1 plant will be dwarf. So, which ratio are we getting? We are getting 3:1 ratio. Out of 4, 3 tall and 1 short. And what will be the genotype that I told you? Right? Now, here 25. Okay, 25. Suppose out of 100 plants, 25 will be purely tall. As such, 75 are purely tall, but if I talk about the genotype, 25 plants will be having capital T, and 50 plants will be having capital T, small t, and 25 plants will be having small t, small t. Okay, this is monohybrid cross. Now, moving on to the next law, law of independent assortment. Mendel's second law of inheritance or law of independent assortment states that in the inheritance of more than one pair of traits in a cross simultaneously, okay, the factors responsible for each pair of traits are distributed independently to the gametes. Now, here children, what they are trying to convey, if you see this, you know, for example, this is dihybrid cross because we are considering 2 characteristics. So, out of 7 characteristics, they can ask for any 2 characteristics. You can take any 2 characteristics and you can explain this law, the law of independent assortment. Okay. So, here if you see this, the seed yellow in color, okay, and pure breeding round and yellow colored seed. At the same time, we have pure breeding wrinkled and green seeds. Can you see here? Right? And then the gametes, parental gametes here will be capital R, capital Y, small y, because these 2 are recessive traits, these 2 are dominant traits. Now, when you do cross pollination between them, you will get a seed which is having yellow color and round shape. That means we are going to observe dominant trait, right? So, what will be the genotype? It will be capital R, capital Y, small y. Previously, it was, can you recollect, capital R, capital Y, capital Y. And in the recessive one, small r, small y, small y. Now, self-pollination, okay. Now, it is self-pollination and we will discuss this in the next slide. Please children, I hope you have understood till here. Very nicely they have explained. You are supposed to make this. It is also mentioned on the page number 145 also, but they have not mentioned it in detail, but you are supposed to learn it in detail. Okay. Okay, now see here what will happen. Mendel selected pea plants having two pairs of characters, shape and color of seed. He selected plants having round yellow seeds and wrinkled green seeds, as we have discussed in the previous slide, and cross-pollinated them. He obtained all plants with round yellow seeds. Why? Because these are the dominant traits in F1 generation. When these plants were self-pollinated in F2 generation, out of 16 plants, 9 had round yellow, 3 had round green, 3 had wrinkled yellow and 1 had wrinkled green seed in the ratio 9:3:3:1. Now children, here you have to sit with your bio notebook and you will be copying this. I will tell you what you are supposed to copy. You can see here parental generation. Can you see this capital R, capital Y, capital Y, small r, small y. Then all will be what? F1 generation, here capital R, capital Y, small y. Now you have to take the gametes from it. What will be the gametes, possible gametes? You can see here four arrows. Can you see? Yes. Capital R, capital Y, 4 combinations. Tell me, capital R, capital Y, capital R, small r, capital Y, small r, small y. In the same way, here also place these gametes. These are the male gametes, these are the female gametes. And now here you have to find out what you will get. So just catch hold of capital R, capital Y and write here. So capital R, capital Y, capital Y. Can you see, children? Here also just catch hold of this. Okay, just circle it. Capital R, capital Y, small y. Can you see here? Yes. Capital R, small r, capital Y. Can you see? Third one? Yes. Then we have capital R, small r, capital Y, small y. Are you getting my point? So wherever we have capital R, the seed will be round. Wherever we have capital Y, the seed will be yellow in color. Now catch hold of this capital R and small y. Can you see in the slide? All of you please concentrate here only. So here then capital R, capital Y, small y, right? Then we have capital R, okay, and small y. That means it is round, shape is round, but now it is green in color because both are small y. Then we have here capital R, small r, capital Y, small y. Can you see? They are round in shape, yellow in color. Again remain along with this line only. Capital R, small y, small y. Can you see now? Round shape, but the color will be green. Now come to the third line. Now small r, capital Y, catch hold of this. Now cross with this, always write capital letters first. So capital R, small r, capital Y, right? Round and yellow. Then we have capital R, small r, capital Y, small y, round and yellow. Then we have small r. Can you see small r, small? Yes. And then we have capital Y. It will be wrinkled and yellow in color. Okay. Then we have small r, small y. But before that you write capital Y and small y. That means RR means here it will be wrinkled, okay, and yellow in color. Move to the fourth line. Small r. First of all you will write capital R, small r, capital Y, small y, round and yellow. Then we have capital R, small y. Okay, yes, it will be round and green, right? Then we have small r, capital Y, small y. It will be wrinkled and yellow. Then we have small r, small y. Can you see, children, here? It will be wrinkled and it will be green in color also. Now what are the resulting genotypes? You can see 16 plants, total 16 blocks are there. We are calling it as Punnett square. 9 will be round and yellow. Can you count? 9 seeds, 1, 2, 3, 4, 5, 6, 7, 8, 9. These are 9 will be round and yellow. 3 will be round and green. You can see in this picture. And then 3 had wrinkled and yellow. These 3 are wrinkled and yellow. And only 1 will be having green seed, and that too wrinkled one. Okay, and which ratio are we going to get? We are going to get 9:3:3:1. This is the phenotype. Remember this, which we can see. Genotype will be a long chain, you have to write all these genotypes, remember this. So here we are talking about the phenotype in F2 generation. In F1 generation it will be what? All plants will be round and yellow only, right? You can see in this slide also, the same slide here, R is equal to dominant allele for seed shape, that's round. Small r is recessive allele for seed shape, wrinkled. Capital Y is dominant allele for seed color, that's yellow, and small y is recessive allele for seed color, that's green. Okay, remember this, you are supposed to draw this. This is Punnett square, and in dihybrid cross we are going to get 16. In monohybrid cross we will be getting only 4. Okay. So in today's class we have discussed about the monohybrid cross, where we took example of the tall plant and the dwarf plant, and in the next section of the slide we discussed about the dihybrid cross. At the same time we discussed about the Mendels law of independent assortment and law of segregation. Along with that we have discussed about the law of dominance. I hope you have understood. You need to practice it once and twice for monohybrid as well as dihybrid.